Hi Emma. What exactly is light microscopy and what's its history?
Light microscopy is a technique for viewing and evaluating structures using transmitted or reflected light. It all started back in 1590 with Zacharias Janssen, a lens maker in Middelburg. Later, in 1665, Robert Hooke provided the first detailed account of the microscope, introducing the term cell, which means small room in Latin. Interestingly, Anton van Leeuwenhoek, a self-taught scientist, took things further by crafting over 500 lenses in his lifetime, becoming a pioneer of microbiology. The industrial production of microscopes began in 1847 with the Zeiss Company.
How does the light microscope work? And what kind of light does it use?
The light microscope uses visible white light, which is essentially a stream of photons with an average wavelength of about 550 nanometers. Human eyes can detect light in the range of 400 to 750 nanometers. The quality of the microscope heavily depends on the lenses and its ability to capture images. With light microscopes, you see a magnified, inverted, and virtual image of the specimen.
What is resolution, and how can we improve it?
Resolution in a microscope refers to its ability to distinguish between 2 points as separate. It's calculated by the formula equals lambda slash d equals lambda slash na, where d is the smallest distance between two distinguishable points. Lambda is the wavelength of the light, and na is the numerical aperture of the lens. The smaller the value of d, the better the resolution. So, reducing the wavelength, for example, by using a blue light filter or increasing the numerical aperture can enhance resolution. We also use immersion oil to improve it further. This oil, with a higher refractive index than air, allows more light rays to enter the objective lens.
How does immersion oil help exactly?
Immersion oil is placed between the lens and the slide to prevent light loss. When light passes through different mediums, like from the air to glass, some of it bends away and doesn't reach the objective. Immersion oil, with a refractive index close to that of glass, reduces this bending, allowing more light into the lens and thus improving clarity and resolution.
What about magnification? How is that calculated?
Magnification is the product of the power of the objective lens and the eyepiece lens. For example, if you're using a 40 times objective lens and a 10 times eyepiece, the total magnification will be 400 times. But keep in mind that useful magnification, where you can still see details, tops out at around 1,000 times for light microscopes. Anything beyond that is called empty magnification, where the image just becomes blurry.
What techniques can we use in microscopy?
Various techniques enhance what we can observe under a light microscope. Bright field. This is the most common and straightforward technique, where light passes directly through the specimen. Dark field. This highlights surface structures like cilia and flagella by blocking direct light, illuminating only the scattered light. Phase contrast. Great for visualizing live cells and tissue cultures as it amplifies slight differences in refractive indices. Polarization. Ideal for studying materials with a crystalline structure, like bone tissue. Fluorescence microscopy. Here, we use UV light to visualize structures tagged with fluorescent antibodies. Electron microscopy. Though it's not technically a light microscopy technique, it offers the highest resolution for observing the tiniest particles, using electrons instead of light.
What if we wanted to look at the smallest possible structures, like viruses or DNA? Could we still use a light microscope?
That would be pushing the limits of light microscopy. The resolving power of light microscopes is around 200 nanometers, which means you won't be able to see structures like viruses or DNA in detail. For that, you'd need electron microscopy, which has a much higher resolving power.
Thank you for listening. If our podcast has been valuable to you, we would greatly appreciate it if you could leave us a 5-star rating and write a review on Spotify and Apple Podcasts, or subscribe, like, and share on our YouTube channel. Your support helps us greatly with discoverability. We hope to see you soon.
Hi Emma. What are prokaryotic cells, and how do they differ from other cells?
Prokaryotic cells, like bacteria and cyanobacteria, are unique in that they don't have membrane-bound organelles. That means there's no nucleus or other complex structures like mitochondria or the endoplasmic reticulum. They're quite small, usually between 1 to 5 micrometers in size. You'll find them in various shapes like spheres, rods, or spirals. Cyanobacteria are especially important because they produce oxygen through photosynthesis.
Do all prokaryotes have the same structure?
Generally, yes, but there are some differences. For instance, archaea, which are also prokaryotes, have a unique cell wall composition. Antibiotics that target bacterial cell walls don't work on archaea because their cell walls are made of different materials. Interestingly, archaea share some features with eukaryotes, such as the way they handle genetic information. You can often find them in extreme environments, like very hot, salty, or acidic places.
Can you tell me more about the bacterial cell wall?
Sure. The bacterial cell wall is mainly made of a substance called peptidoglycan, which is a mesh-like structure composed of sugar derivatives and amino acids. The sugars, N-acetylglucosamine, Ng, and N-acetylmuramic acid, Nm, are linked together, and short peptide chains connect these sugar molecules. An enzyme called transpeptidase forms cross-links between the peptides, strengthening the wall.
How do we classify bacteria based on their cell walls?
We use a technique called gram staining. It helps to distinguish between two major types of bacteria, gram-positive and gram-negative. Gram-positive bacteria have a thick peptidoglycan layer that retains a violet dye, so they appear purple or blue under the microscope. Gram-negative bacteria, on the other hand, have a thinner peptidoglycan layer and an additional outer membrane, which doesn't retain the violet dye. Instead, they appear red or pink after being stained with a counterstain like safranine.
What's the role of plasmids in bacteria?
Plasmids are small, circular pieces of DNA that replicate independently of the bacterial chromosome. They often carry genes that give bacteria an advantage, like antibiotic resistance, the ability to produce toxins, or fix nitrogen from the atmosphere. In genetic engineering, plasmids are used as vectors to insert specific genes into bacteria, which can then produce desired proteins.
How do bacteria move?
Many bacteria use flagella, which are long, whip-like structures that help them move. Some also have pili, which can help with movement but are mainly used for attachment to surfaces or other cells. Pili are also involved in a process called conjugation, where bacteria exchange genetic material.
What's sporulation?
Sporulation is a survival mechanism used by some bacteria. They form spores, which are highly resistant structures that can survive in harsh conditions, such as extreme heat or dryness. These spores can remain dormant for years, and when the conditions improve, they can reactivate and cause infections.
What about antibiotics? How do they work?
Antibiotics are chemicals that can kill or inhibit the growth of bacteria. They work by targeting essential bacterial processes like cell wall synthesis, protein production, or DNA replication. For example, penicillin inhibits the enzyme that forms crosslinks in the bacterial cell wall, causing the bacteria to die. However, overuse of antibiotics has led to resistance, where bacteria evolved to survive antibiotic treatment.
What happens when bacteria become resistant to antibiotics?
When bacteria become resistant, it's much harder to treat infections. Resistance can develop due to the misuse of antibiotics, like not finishing a prescribed course or using antibiotics for viral infections. Some bacteria acquire resistance through horizontal gene transfer, where they receive resistant genes from other bacteria. These resistant bacteria can spread, making infections more difficult to control.
Are there alternatives to antibiotics?
Yes. Researchers are exploring alternatives, like antimicrobial peptides, AMPs, which are natural components of the immune system. These peptides can kill bacteria by disrupting their membranes. Other options include bacteriophage therapy, which uses viruses that specifically infect and kill bacteria, and certain medicinal plants with natural antibiotic properties.
Thank you for listening. If our podcast has been valuable to you, we would greatly appreciate it if you could leave us a 5-star rating and write a review on Spotify and Apple Podcasts, or subscribe, like, and share on our YouTube channel. Your support helps us greatly with discoverability. We hope to see you soon.
Hi, Emma. What are the primary differences between plant and animal cells?
Great question. Both plant and animal cells are types of eukaryotic cells, meaning they have a nucleus and other membrane-bound organelles. However, they do have key differences. Let's start with size. Plant cells typically range from 10 to 100 micrometers, while animal cells are usually between 5 and 50 micrometers.
So, what makes up the structure of a plant cell?
In plant cells, the structure is strongly defined by the cell wall, which is made up of cellulose and sometimes other components like hemicellulose and pectins. The cell wall gives the cell its shape and supports the entire plant structure. Additionally, plants have something called plasmodesmata, which are small channels that allow for communication and transport of substances between cells.
I've also heard about chloroplasts in plant cells. Can you explain those?
Absolutely. Chloroplasts are unique to plant cells and are where photosynthesis occurs. They contain their own DNA and are thought to have originated from a symbiotic relationship with cyanobacteria. This is a great example of the endosymbiotic theory, where an early eukaryotic cell took in a photosynthetic organism, leading to the evolution of chloroplasts.
And what about mitochondria? Do both plant and animal cells have them?
Yes, mitochondria are found in both plant and animal cells. They're the powerhouses of the cell, generating energy through cellular respiration. They also have their own DNA, supporting the theory that they, too, originated from an ancient symbiotic relationship.
What are the unique features of animal cells, then?
Animal cells have structures called lysosomes, which are enzyme-filled organelles responsible for breaking down waste materials within the cell. Additionally, animal cells have a flexible plasma membrane that contains cholesterol, giving it fluidity and allowing the cell to change shape as needed. Unlike plant cells, animal cells don't have a rigid cell wall, which gives them a more flexible, often rounded shape.
I see. And how do plant and animal cells divide?
Good observation. Plant cells divide by forming a cell plate, also known as a phragmoplast, which grows outward until it splits the cell into two. Animal cells, on the other hand, divide through a process called cytokinesis, where the cell membrane pinches inward, eventually splitting the cell in two.
So, what are some of the foundational differences between plant and animal cells?
That's a good starting point. Both plant and animal cells are eukaryotic, meaning they have complex structures, including a nucleus. However, there are significant differences. Let's start with plant cells. They're typically larger, around 10 to 100 micrometers, while animal cells range between 5 and 50 micrometers.
Interesting. What makes plant cells so unique?
Plant cells have a rigid cell wall made primarily of cellulose, but it can also include other materials, such as hemicellulose and pectins. This wall gives structural support and helps the plant maintain its shape. Plants also have something called middle lamella, a pectin-rich layer that acts as a glue between adjacent plant cells.
I've read about different types of plant tissues too. How are these structured?
Exactly. Plant tissues are made up of various cell types that each play specific roles. Parenchyma, for instance, is a type of plant tissue that's involved in functions like photosynthesis, storage, and regeneration. Then there's collenchyma, which provides structural support, especially in areas of the plant that are still growing. Another type, sclerenchyma, is very rigid and helps support mature parts of the plant, and it's strengthened by a secondary cell wall.
And what about those chloroplasts? I know they're important for photosynthesis.
Yes, chloroplasts are fascinating organelles. They are semi-autonomous, meaning they contain their own DNA and can replicate independently. Chloroplasts are where photosynthesis occurs, converting light energy into chemical energy stored in glucose. Interestingly, these chloroplasts are thought to have originated from an ancient symbiosis with cyanobacteria.
So, they were once independent organisms?
That's the idea. It's based on the endosymbiotic theory. This theory suggests that certain organelles, like chloroplasts and mitochondria, originated from free-living bacteria that were absorbed by early eukaryotic cells. Over time, they became integral parts of the cell.
Speaking of mitochondria, both plant and animal cells have them, right?
Correct. Mitochondria are known as the powerhouses of the cell because they generate energy through cellular respiration. They are also semi-autonomous with their own DNA, just like chloroplasts. Plant cells use mitochondria to produce energy, especially in areas where photosynthesis can't meet the cell's energy demands.
Do animal cells have any unique structures?
They certainly do. Animal cells lack a cell wall, which gives them flexibility. They do have lysosomes, which contain enzymes to break down waste materials through a process called hydrolysis. Animal cells also contain centrioles within a structure called the centrosome, which is essential for cell division.
So, centrioles are crucial for cell division?
In animal cells, yes. Centrioles organize microtubules during cell division, helping to separate chromosomes accurately. However, most plant cells don't have centrioles. Instead, they rely on other microtubule organizing centers, showing that centrioles aren't strictly necessary for cell growth.
I've heard that plant and animal cells respond differently to their environments. Can you explain that?
Great point. In a hypertonic environment, where there's a high concentration of solutes outside the cell, animal cells shrink in a process called plasmaryza, whereas plant cells retain their rigid shape because of the cell wall. However, the living part inside the plant cell, called the protoplast, will shrink away from the wall, a process called plasmolysis. In a hypotonic environment, where water concentration is higher outside the cell, an animal cell may burst due to osmotic pressure. Plant cells, however, can take in water and swell, but the cell wall prevents them from bursting. This pressure buildup, known as turgor, helps maintain plant structure.
That's a lot of information. What about the extracellular matrix? How does that fit in?
The extracellular matrix, or ECM, is essentially the noncellular component present within tissues and organs. It's particularly important in animal cells, where it provides structural support and facilitates cell communication. ECM is rich in proteins like collagen, which provides tensile strength, and proteoglycans, which retain water and form a gel-like structure that supports cellular adhesion.
So, ECM is mostly for structural purposes?
Not solely. ECM has diverse functions. For example, it plays a role in cell migration, wound healing, and even metastasis of cancer cells. Certain proteins in the ECM, called integrins, anchor cells to their surroundings and enable communication. When these integrins bind to specific ligands in the ECM, it triggers a signal cascade within the cell, influencing processes like adhesion, migration, and endocytosis.
Are there any conditions linked to the ECM?
Absolutely. ECM-related pathologies often arise from mutations in genes that code for its components. For instance, defects in collagen formation can lead to connective tissue disorders. Osteogenesis imperfecta, or brittle bone disease, is one such condition caused by mutations in collagen type I. Another example is Ehlers-Danlos syndrome, where faulty collagen results in overly elastic skin and hypermobile joints.
And what's the role of elastin in the ECM?
Elastin is another protein found in the ECM, especially in tissues that need to stretch and then return to their original shape, like skin, blood vessels, and lungs. Elastin molecules allow tissues to maintain elasticity, which is vital for organs that frequently expand and contract.
Fascinating. Does ECM have any role in the immune response?
Yes, indirectly. For instance, certain ECM components, like fibronectin, act as scaffolds for cells during wound healing. Fibroblasts are cells that produce ECM, and in cases of tissue injury, they are particularly active, producing more ECM to support the healing process.
What happens if there's a mutation in integrins or other ECM-related proteins?
Mutations in integrins or other ECM components can lead to inadequate cell adhesion, which might cause cells to detach too easily. This is a mechanism behind certain cancer cells that lose their attachment to primary tumors and travel to other parts of the body. Additionally, defective ECM interactions can impair wound healing or immune cell function.
Thank you for listening. If our podcast has been valuable to you, we would greatly appreciate it if you could leave us a 5-star rating and write a review on Spotify and Apple Podcasts, or subscribe, like, and share on our YouTube channel. Your support helps us greatly with discoverability. We hope to see you soon.
Hi Emma, I've been reading about microscopy techniques, but I'm curious. Why do we use electron microscopy?
Excellent question. We use electron microscopy, or EM, to study structures within cells that are too small to see with light microscopes. Electron microscopes allow us to visualize cell organelles, macromolecules, viruses, and even subcellular structures in incredible detail.
How does it work? What makes it so powerful?
Electron microscopes use a stream of electrons instead of light. This stream is generated by an electron gun, which produces a beam that behaves similarly to light but with a much shorter wavelength. Because of this short wavelength, electron microscopes have a very high resolving power, allowing us to see details on the nanometer scale. In fact, depending on the technique, EM can achieve magnifications of up to 1,000,000 times.
So, what's the difference between electron microscopes and light microscopes?
Light microscopes use glass lenses to focus light, while electron microscopes use electromagnetic lenses to manipulate the electron beam. EM images are captured in black and white, although color can be digitally added later to highlight specific structures.
I've heard there are different types of electron microscopes. What are they?
There are two main types: transmission electron microscopy, TM, and scanning electron microscopy, SEM. Each one has distinct capabilities. Let's start with TM.
Sure. What does TM do?
TEM is designed to observe internal structures within cells. In TM, electrons are transmitted through an ultra-thin specimen, usually less than 500 nanometers thick. The electron beam passes through the sample, creating a highly detailed 2D image of the internal structures.
And how are these samples prepared?
Preparing samples for TM is meticulous. The samples need to be carefully dehydrated, as water interferes with the electron beam, and they are often fixed using chemical or cryo methods to preserve structures. They're also coated with heavy metals like gold or platinum to improve contrast.
What's the magnification like with TM?
TEM can magnify up to 1,000,000 times, with a resolution down to 0.1 nanometers. It's highly effective for studying organelles, proteins, and even viral particles within cells.
What about SEM? How is it different?
SM is excellent for examining surface structures. Unlike TM, which looks inside, SEM scans the surface by bouncing electrons off the sample's exterior. This gives a 3D image of the sample surface, which is particularly helpful for looking at the textures and shapes of biological and non-biological materials.
That sounds useful for many fields.
It is. SEM is popular because sample preparation is relatively straightforward. Samples are coated with a thin metal layer, often gold, to improve electron reflection. With SEM, we get magnifications up to 1,000,000 times, with resolutions around 0.5 to 1 nanometer.
Moving on to another topic. What about vesicular transport? I read that it's essential for cell function.
Yes, vesicular transport is crucial. It's how cells move materials in and out. We can break it down into two main processes: exocytosis and endocytosis.
Could you explain each one?
Certainly. Exocytosis is how cells expel materials. For example, cells constantly produce ECM proteins, digestive enzymes, and hormones. In exocytosis, vesicles containing these materials merge with the cell membrane, releasing their contents outside the cell.
So, exocytosis is like the cell spitting out what it doesn't need?
Exactly. Then we have endocytosis, the process by which cells take in substances. There are different types: pinocytosis, or cell drinking, where the cell takes in fluids, and phagocytosis, or cell eating, where it engulfs larger particles like debris or microorganisms. Another type, receptor-mediated endocytosis, is very specific. It allows the cell to absorb targeted molecules like certain hormones or nutrients.
I've heard about transcytosis too. How does that fit in?
Transcytosis is fascinating. It's a specialized process where substances are transported across the entire cell. This happens in cells with a high rate of material exchange, like in the lining of the intestines. The substance is absorbed via endocytosis, transported through the cell, and then released on the opposite side via exocytosis.
Let's shift to the cytoskeleton. What are its components?
The cytoskeleton is the cell's structural framework. It has three main components: microtubules, microfilaments, and intermediate filaments.
I'm familiar with microtubules. They're involved in cell division, right?
Yes, microtubules are made of tubulin proteins and play critical roles in cell division and transport. They form a structure called the mitotic spindle, which helps divide chromosomes during cell division.
And microfilaments? Are they similar?
Microfilaments are thinner and more flexible. They're primarily composed of actin, which interacts with another protein called myosin. This interaction allows the cell to contract and move, making microfilaments essential for cell motility and shape.
What about intermediate filaments?
Intermediate filaments provide tensile strength, helping the cell resist mechanical stress. They're more stable than microtubules and microfilaments and form a supportive network throughout the cell.
I've also read about molecular motors. How do they work with the cytoskeleton?
Molecular motors are proteins that move along the cytoskeleton, powered by ATP. There are three main types: myosin, kinesins, and dyneins.
What's the role of each type?
Myosin move along actin filaments and are crucial for muscle contraction and transport within cells. Kinesins and dyneins travel along microtubules but move in opposite directions. Kinesins typically move toward the plus end, helping transport organelles and vesicles toward the cell's outer regions. Dyneins move toward the minus end, often carrying materials inward and playing a role in the movement of cilia and flagella.
It's incredible how much goes on inside a single cell. Thank you for explaining all of this in such detail.
You're very welcome. Electron microscopy, vesicular transport, and the cytoskeleton all reveal the cell's complexity, helping us understand everything from basic cellular structure to how diseases develop at the microscopic level.
Thank you for listening. If our podcast has been valuable to you, we would greatly appreciate it if you could leave us a 5-star rating and write a review on Spotify and Apple Podcasts, or subscribe, like, and share on our YouTube channel. Your support helps us greatly with discoverability. We hope to see you soon.
Hi Emma, could you explain what a model organism is and why it's so important in research?
Certainly. A model organism is one that researchers study intensively to understand biological principles that can apply to other organisms as well. These organisms often have specific traits that make them ideal for research. They typically have short life cycles, produce large numbers of offspring, are economical to maintain, and have fully sequenced genomes. These characteristics allow us to generalize findings across different species. Examples of model organisms include bacteria, yeast, frogs, mice, and even plants like corn.
Interesting. Now, what about tissue and cell cultures? How are they used in research?
Tissue and cell cultures allow us to study human cells in a controlled environment without needing a full organism, which is essential for ethical reasons. This approach, known as in vitro, involves cultivating cells outside their natural environment under precisely defined conditions. We use these cultures for various purposes, including drug development, genetic studies, and even prenatal diagnostics.
And what makes this method advantageous?
Using cell cultures has multiple benefits. It allows us to get results quickly and control every factor in the cell's environment, which isn't possible in a whole organism. Additionally, since these cells are outside the body, we don't face the ethical issues of experimenting directly on humans. Cultures are also quite accessible for genetic manipulation.
Are there any downsides?
Yes, there are. Cells in a culture lose the context of the whole organism, so we miss complex interactions that happen within a living body. Additionally, there's a risk of contamination, and over time, cultured cells can accumulate mutations, altering their properties.
That makes sense. So, how do you set up a cell culture?
When we start a culture, we need to transfer tissue into a nutrient solution as quickly as possible to mimic the body's environment. We control various factors like salt concentration, pH, and temperature. A laminar flow cabinet sterilizes the workspace to prevent contamination. We use a growth medium, usually with serum, nutrients, and growth factors, to sustain the cells. Equipment like CO2 incubators, inverted microscopes, and centrifuges are also essential for observing and maintaining the culture.
What are the main types of cell and tissue cultures?
There are two main categories: tissue cultures, where cells remain attached and interact, and cell cultures, where cells are isolated. Primary cultures come directly from an organ and contain a mix of cell types. They don't survive long but can be passed or passaged into a new medium. Secondary cultures are derived from primary cultures and can survive through multiple passages, though they still have a limited lifespan due to the Hayflick limit.
What about cell lines? I've heard that some cells can divide indefinitely.
Right, cell lines are what we call immortal, because they can divide without limit. This can occur naturally, as in cancer cells, or by introducing specific mutations. A famous example is the HeLa cell line, derived from a cervical cancer patient named Henrietta Lacks in 1951. HeLa cells have been crucial in developing the polio vaccine, cancer research, and even space experiments.
Speaking of cell growth, what are the stages cells go through in culture?
Cells in culture progress through several growth stages. Initially, there's a lag phase, where they adapt to the new environment. This is followed by the log phase, where they grow exponentially. In the stationary phase, growth halts due to nutrient depletion and toxin accumulation. Finally, if conditions aren't improved, they enter the death phase, where cells start to die off.
I've read about stem cells, too. Could you describe their role in research and medicine?
Certainly. Stem cells are unique because they're undifferentiated, meaning they can develop into various cell types. Embryonic stem cells are pluripotent, capable of becoming almost any cell type in the body, while adult stem cells, like those from bone marrow, are multipotent and more limited. We use stem cells for regenerative medicine, repairing damaged tissues, treating neurodegenerative diseases, or even reconstructing organs.
That's fascinating. And is there a special material that's used to grow cells in 3 dimensions?
Yes, that would be matrigel. It's a protein-rich, gelatinous substance secreted by mouse tumor cells, used to mimic the extracellular environment. At low temperatures, it's viscous, but at body temperature, it forms a gel that allows cells to grow in 3D, simulating conditions in a living organism. It's invaluable for studying cancer, stem cell differentiation, and drug testing.
Thank you. This really clarifies how cell and tissue cultures are used in research and medicine. What does it mean to passage cells in culture? Is it a common process?
Yes, passaging is essential for keeping cell cultures viable over time. As cells grow and cover about 70% of the culture dish, they use up nutrients and space. We then transfer some of these cells to a fresh medium in a new dish. This prevents overcrowding, called confluence, which would otherwise halt cell division and lead to cell death if not managed.
Are there special steps involved in passaging?
There are. First, we remove the old medium, wash the cells with a phosphate-buffered saline solution to remove residual nutrients, and add an enzyme called trypsin. Trypsin detaches the cells from the dish, allowing us to collect and transfer them. We then centrifuge them, removing any enzyme remnants, and resuspend them in fresh medium.
I've heard cells can be frozen for long-term storage. How does that work?
Correct. This process is called cryopreservation. We cool the cells very slowly, often using controlled-rate freezers to prevent ice crystal formation that could damage the cells. A substance called dimethyl sulfoxide, DMSO, acts as a cryoprotectant, preventing crystallization. Once frozen, the cells are stored in liquid nitrogen. When needed, we thaw them quickly, reintroducing them to the culture under optimal conditions.
That's incredible. So, what about the growth cycles you mentioned earlier?
Cells follow specific growth phases. Initially, in the lag phase, they adapt to the new environment. Then comes the log phase, or exponential growth, where they rapidly multiply, consuming nutrients and producing byproducts. Once these nutrients deplete, the cells reach the stationary phase where growth stops. Without intervention, they enter the death phase, where cells start to die as the conditions become inhospitable.
Are there any specific tools or equipment required to handle these cell cultures?
Absolutely. Besides the growth medium and controlled environment, essential tools include a laminar flow cabinet to maintain sterility, an incubator to control CO2 and temperature, an inverted microscope for detailed observation, and a centrifuge for processing cells. These tools help create and maintain the ideal conditions for cell survival and growth.
Understood. Now, what's the role of stem cells in research?
Stem cells are key players in regenerative medicine due to their ability to differentiate into various cell types. Embryonic stem cells, derived from early embryos, are pluripotent, meaning they can become any cell type in the body. Adult stem cells, found in organs like bone marrow, are multipotent and are more limited, generally forming cell types within their tissue of origin. We're exploring their applications for repairing tissues, treating neurological conditions, and rebuilding damaged organs.
Is there anything unique about embryonic stem cells?
Embryonic stem cells are indeed unique because of their pluripotent capacity. They're often derived from early-stage embryos created via in vitro fertilization, where unused embryos are either stored, discarded, or sometimes used for research. These cells offer immense potential for therapeutic cloning and regenerative treatments, although ethical considerations guide their use.
And what are totipotent, pluripotent, and multipotent stem cells?
Good question. Totipotent stem cells, found in early embryos, can develop into a complete organism, forming any cell type, including those in the placenta. Pluripotent cells can develop into almost any cell in the body, but can't form an entire organism. Multipotent cells, like those in adult tissues, have a more limited scope, differentiating only into cell types within a specific tissue or organ.
Lastly, I'm curious, what challenges do researchers face with embryonic stem cells?
With embryonic stem cells, there are challenges on multiple fronts. Ethical concerns often arise, given their source from early embryos. Also, controlling their differentiation precisely can be complex. However, when managed well, they're a powerful resource, enabling groundbreaking advances in treating various conditions and exploring therapeutic cloning.
This has been incredibly enlightening. Thank you for such a thorough overview of tissue cultures and stem cells.
Thank you for listening. If our podcast has been valuable to you, we would greatly appreciate it if you could leave us a 5-star rating and write a review on Spotify and Apple Podcasts, or subscribe, like, and share on our YouTube channel. Your support helps us greatly with discoverability. We hope to see you soon.